And there are some of the other cases in coding genes itself to be transferred. The cases in RPS2 and RPS11 in mitochondria of plants. It has been documented a couple years ago, and again, it was detected by Southern blot and also the comparator phylogeny. And the original Southern blot shows this kind of very special pattern for RPS2 among the different plant species. So you can imagine that their lab did a lot of Southern blot, just like the last case. So they want to see if there's any interesting patterns among the different mitochondrial genes. And they found RPS2 was absent from many of the plant species, and there are also some missing part over here. So again, we can map the pattern onto a known species tree. And so in this case, this is RPS2, and on the right there's RPS11. So the color of this branch showing this present or absent of the RPS2. So this is the tree from somewhere else. So it's again considered as a species tree. So this is a known tree, and the red lines indicates the presence of the RPS2. So there are so you can imagine that the basal angiosperm and throughout the evolution, there's always have RPS2, but was missing here, 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 and this whole clade are missing. And somehow there's some presence of RPS2 in Actinidia, that's kiwi. So how to explain that? There are two ways, of course, and one is there are multiple losses, and among all the different losses. So originally it still has that, but only Actinidia still retain that, and there are a lot of parallel losses among this whole group, which is very unlikely. And the other would be that the sequence of Actinidia RPS2 was horizontally transferred from somewhere else. So that's likely. And the RPS11 is also showing the similar case that Lonicera and Batua, these two taxa, are present for RPS11. So the next step would be to reconstruct the phylogeny using these two sequences. Okay, well, this is just the model we just mentioned. And of course, there's one possibility that there's intracellular gene transfer of the particular genes to nuclear genome. And this usually happened long time ago for the mitochondrial gene to nuclear. But in plant, since in the case of chloroplast, the chloroplast genes are transferred to nuclear still continuously, even now. So this is still possible. And if this is the case, then you can explain the Southern blot pattern, because in this, if you realize the pattern of the Southern blot, you will realize that because if a particular gene is in nuclear, and if they are still in organelle, they will be quite different, since that usually you will have 50 copies of organelle compared to 1 copy in nuclear genome. So the signal will be 50 to 100 to 1. And if you see the signal here very, very bright, then in this case, it means that it's still within the organelle genome. But it's sometimes, if it's missing here, doesn't mean it's totally lost. It could be transferred to nuclear. But the signal will be very, very light. And this because of the Southern blot pattern. And the other would be the two explanation I just mentioned. There is could be a lot of massive losses in U die-cast, or there's a horizontal gene transfer. So we can what we can do is we can reconstruct the phylogeny. So we reconstruct the phylogeny using the mitochondrial RPS2. And based on RPS2, actinidia will be grouped. Actinidia here will be grouped with these red lines, which are monocots. And we know that kiwi is not a monocot. Kiwi is somewhere here. So the best explanation is there's a horizontal gene transfer from monocot RPS2 to actinidia and replace the one in actinidia mitochondria. Similar case for RPS11. You will see this is the expected position for Lonicera, but it's grouped with several others, which is in Reniculitis and Reniculitis malgan. And so that means that the RPS11 was transferred from Reniculitis, which is also very unique if you consider how's that possible. And there's another case in Sanguinaria, and in Sanguinaria is even more extreme in that if you compare the sequence of RPS11 in Sanguinaria, is present, but if you cut in half and then put into the phylogenetic analysis, the 5' half will show you the Sanguinaria group with this blue thing, and the 3' half of this Sanguinaria will group with the monocots, which means that there's some 3' half of the Sanguinaria was from the monocots, and then create a chimeric here. And the evidence is very pronounced in that if you compare the sequence, it's right here in the middle, and the 5' half is very similar to something else, and 3' half is similar to the monocots. And these particular genes, RPS11 in Sanguinaria, is expressed. So if you do the RT-PCR, it's there, and also it is transcribed. Of course, we still have no idea if it's functional because we have no data on the protein sequence as well. And for this RT-PCR data, and there are several other cases of HGT, also show that the new gene or the horizontal gene transfer gene still could be transcribed. But in most other cases, or I should say, half of the cases for these HGT genes are pseudogenes. So this is the basic scheme for mitochondrial genes, and probably need to talk about it. Okay, this is the Amborella cases for ATP1, and this is also kind of interesting. I can talk a little bit more. In 1999, there's one sequence of ATP1 from Amborella was sequenced and then put into phylogeny, and they found a group with U die-cast, which is not possible since Amborella is a basal angiosperm. And Qiu Yinglong, and he explained this is due to a long branch attraction to attract the Amborella sequences to the other U die-cast, which is possible, but we have no idea. But Bachman, they identified another copy of the Amborella ATP1 and found out there are actually two copies of ATP1 in Amborella. And this one is occupied at a dry position for Amborella as a basal angiosperm, So this means that another copy, a parallel of ATP1 of umbrella was from somewhere else, in this case in U die-cast. So this is kind of a small summary for the HDT events in angiosperms happen here. So there are several different cases between from monocots to like actinidia or to sanguinaria, and also other cases from the estuaries to umbrella, from U die-cast to umbrella. So how does this happen? Since the cases on this figure does not involve any of the parasitic plant at all, so it means that it must through other cases, other mechanisms, and we just mentioned several mechanisms in previous slide. Parasitism is one, as hybridization is possible, but it's very unlikely to be hybridized. This is on a different order, so it's very unlikely they could hybridize. And so it means that it could be only be transferred by some particular vectors through like a virus or fungi is also possible. So that's the only explanations that for this mitochondrial transfer, HDGT transfer in here.